Hello and good evening to all the viewers present here. Welcome to the day 7th and final track of Open Pharmacy Federation. Good evening to Dr. Vikram Chaudhary, Director of OPF, Dr. Paresh Dave, Founder of IP Movement, Honorable Speakers Dr. Girinath G Pillai, Mr. B. Venkteshwarulu and Dr. Andrew Tait, and all the viewers to the final day of Pharmanist 21 conference organized by Open Pharmacy Federation. I, Chavi Goyal, along with Dr. Paresh and Valika, are moderators for the today's session. So before starting the session, I hope all are doing fine in these desperate times and holding up with the hope. So starting with the today's session, I would like to let all participants know about the Pharmanist 21. So the theme of the Pharmanist 21 is data science, computational tools, artificial intelligence, novel technologies, evaluation and contribution in the pharmaceutical research. It is an international virtual conference that welcomes personnel from a plethora of industries such as pharmaceutical, regulatory, academia, research, and many others focused on the innovations and betterment of the healthcare system. So these conferences mainly focuses on various aspects such as innovations, current trends, modern discoveries, which will ultimately help in the development of healthcare system. So the main purpose of organizing an international conference is to provide a platform and accessibility to the emerging leaders and the youth of the society to make their future bright. Farminess 21 includes knowledgeable webinars like this one we are holding today. Webinars by the leaders from industries and academia from all over the globe, along with various competitions such as e-blog, quiz, e-poster, video making, extempore, and many more, enabling the participants to get various opportunities to learn and participate in various competitions and win prizes of worth rupees 10.51 lakhs. In addition to this, there is one more golden opportunity from the conference. The winner will get benefit of international exchange program, which means that the selected candidate will get the exposure of learning in international industries and academia with their respective work culture. So I would also like to register here that the participant who haven't registered yet must also do that before we are closing the registrations. So moving on to today's session, I request all the participants to type their questions in the chat box and the speakers will answer them after the session. We have 3 sessions lined up today, so hold your patience for that. And let me introduce you all to Dr. Pillai for machine learning in drug discovery for drug candidate. Dr. Pillai is a coordinator for Drug Discovery Hackathon, research scientist at Nyro Research, and founder of Zestra Innovations Bangalore. He was formerly a Marie Curie Research Fellow at Molcord Limited, Estonia. He carried out his PhD at University of Florida with Professor Carlson and with Professor Karczyński. I hope this is the pronunciation. I'm sorry if it is not. University of Tartu. His work on mosquito repellents with US Department of Agriculture, including SAR and machine learning methodologies with targeted descriptors, attracted global collaborators for scientific findings. He was the responsible researcher for 4 industrial products from Henkel, ExxonMobil, Johnson & Johnson, and Eastman in the US. He holds 37 research papers in peer-reviewed international journals, 2 US patents, and co-authored a book chapter. Dr. Pillai is having expertise in target identification. Currently, he has a PhD student working under him in the area of aging registered under VIT. Now, I would like to hand over the platform to Dr. Pillai. Over to you, sir.
Thank you so much, Chavi, for the kind introduction. And the pronunciation was Professor Katrutsky, Alan Katrutsky. Anyway,
Okay, sir.
that's not a problem. Thank you. So let me share my screen. Could you please confirm whether you're able to see my slide?
Yes, sir, we are able to see, sir.
Great. Thank you so much. So as the time is given, like 30 to 40 minutes, I'll try to take. And before I start, since we are talking about, as we presume, it's a hot topic nowadays, but this was being done back in 2000s or 2010, 2012. But that time we were more talking about statistics rather than calling it as AI or ML. But this particular terminology got too much of attention very recently. Let's go through that. But the main idea of this particular presentation is how this can be used in drug discovery for a drug candidate selection. I did not mention how it could be useful to discover a drug, but about a drug candidate is something very important that we have to look at. I also wanted to have a disclaimer here. Many of the pictures and videos or codes that is being used here as being used with the right to reuse, and the copyrights and ownership goes to the respective authors. There are multiple resources and IDs being used and you can make your own right choice which one you have to implement. And I myself does not declare any conflict of interest and does not have any connection with the employer or employee with this content, and all are my personal views. And slides are also shared via SlideShare. The codes and other workflows are via GitHub and Colab, and detailed videos are also shared via YouTube. I'll be sharing the links once I finish the talk. So it is clearly mentioned that AI is clearly stimulating the drug discovery market and providing new opportunities for companies using such technologies. But the importance of ML technologies, ML is machine learning, AI is artificial intelligence. I know that everybody knows about that, still thought to repeat it. It's likely to increase in the coming years. So it was being said by the scientist in CAD Service Market Report. These are some of the screenshots that I've taken from the drug discovery market report. You can see there are plenty of companies which is looking at different stages, and some of them are very well known to us, and they're also present in India and worldwide. Some are CROs, pharma industry, some are software industry, some are very much expert in the AI or the ML domain. So it is still growing. And cost saving analysis, that's where we are looking at. By implementation of AI or ML, can we reduce the cost of the analysis? And this analysis is being done. All the values are being masked because I did not purchase the report. This was a sample report that I could get it from the website. And the upcoming trends is basically deep learning and machine learning, including cloud-based technology platform. Right now we have AWS, Azure, we also have Colab, even Google Cloud. So similarly, the cloud platforms are going to help us. And artificial intelligence when coming to deep learning and machine learning. But the biggest challenge that we are facing in drug discovery is that scarcity of data or missing data. So data imputation is something very important. Even though we have data, we have to collate it manually or we have to curate it manually. There are several databases available, but for certain objectives of CAD or drug discovery, we still have to sit down and curate our data set. And you can see as the years progressing, the number of CAD service providers being established per year is increasing. So post 2015, you can see more than 25% is being increased. And service provider, which is being distributed by CAD approaches who are implementing CAD. So there you can see a combination where 21 providers are already doing it. And with a combination of ligand-based and structure-based, about 45 providers are there. Fragment based, we have still less number, but it's growing. And there are other providers who mainly concentrate on lead optimization and hit to lead process, hit generation, like virtual screening and other, target identification, target validation. Target identification is the place where too much of machine learning interest is being taken care. Recently, there are few tools that as platforms have been come from AI companies where they are majorly focusing on target identification and validation. But behind this, why do we come up with each of the techniques and why do we want to still go there? Because we know AI is not there much for drug discovery. But for many other areas, yeah, we already have seen its success stories. But still, why we want to go through? Because the cause of failure. The oral drugs have faced cause of failure in early 90s or 80s because of this reason. 40% is almost because of pharmacokinetic or ADME properties, and 28% is almost because of efficacy. But later, When you go on to 2000s or 2010, we could see the trend has changed because SAR, implementation of structure activity relationship, and because of certain principles like REACH, OECD principles, the estimation of ADME at early stages kicked in, and they were able to generate models to predict these properties so that the failures because of this pharmacokinetic or efficacy slightly got reduced. But the biggest problem started with toxicity because we started exploring more different scaffolds or chemotypes. So toxicity came over like 40%, and strategy is a biggest problem because we use certain rules, certain criteria or filtering mechanism or prioritization of certain properties or experimental data to choose your possible lead like molecule in a wrong direction. So strategy also gave us, I'm just trying to tell you in a layman terms. So strategy also gave a major reason why there was failures of oral drugs. That's where we have to see whether the machine learning is really overhyped or not. This was a survey that was being done by CNDN during the year 2018. I know it's already 3 years old, but still useful. So it was being taken to organic chemist, computational chemist, and all. And about 13.9% strongly agree that the machine learning is overhyped, and 31.8% still just agree. But there is again a good amount, like 7.9 and 24.5, they disagree to it. But 21% says that they're not sure about it from 151 respondents. And you can see the major team, the respondents were from organic chemistry and the least were from computational chemistry. But I think if we rerun the survey right now, you could see some increase in the agree and strongly agreeing criteria because we have many packages and libraries available to support in our research. But the impact of AI in CAD, recently there were two great news coming up. One is from in silico medicine, where they identify the drug candidate in weeks and it has entered into clinical trials, right? And also exploring chemical space. Can AI take us where no humans has gone before? And it's already happening, where we can explore a large chemical space, which is more than 7 billion compounds. Can we ever think about it? No. So that's where we are heading to. But in normal life, let's see how machine learning workflows can help in chemistry. Of course, you should have annotated data, including sensors, screening, synthesis, and simulation. And this all data has been achieved from a chemist, of course, through experimental data. We have to pre-process it. Basically, data wrangling, data transformation, data processing. That's what in the machine learning terminologies. And then you get into machine learning approaches or deep learning based upon the category of the data, whether it is a numerical or a continuous or a categorical data, and depending upon the volume of the data. So we look at support vector machine, or also we look at evolutionary and other algorithms. So for example, random forest, or we can always look at decision tree, neural network, even Gaussian process regression methods. So these kinds of different machine learning methods or approaches could be also applied. But what is the main difference between statistical method or models and machine learning models? Because machine learning model has the capability to train the model when you have a new data. But when you have a statistical model, one has to sit down and generate the new model once again. But this can be automated to iteratively process the data when new data comes in to train them. So basically, what is machine learning? You have to learn, make more efficient when it has to be applied. And then we try to classify and based upon the regression or classification. And other way to use chemistry data in ML is that fundamentals of working with active learning algorithm. This is also something that I just mentioned. You have from the based upon the design of experiments, you have a lot of experiments handled, and then you try to generate the model with the measured and the predicted, and then you try to optimize. And then you come up with proposal based upon the prediction of non-experimental designs. And then you try to again add more data, and the model gets self-learned. So active learning process is happening. In this way, you can minimize the number of experiments and maximize the material's properties. Similarly, a framework for working in an in-house database. Let's imagine that you have historical reactions with failures and success stories. Just understanding success stories does not help an ML model, because you need to have failures also in order to have a better prediction capability. So it can be entered from notebooks or e-notebooks and the reactant descriptors, and they try to create a full database of reactions. And then using different models, we will be able to tell you whether the possible reaction that is being processed or proposed is successful or failure. Again, depending upon the data, the predictive capability can be assessed. So no need to say that it is always going to be accurate, completely depending upon the data that you use for your model. And then you can also compare with the data from simulations. For example, you have to do DFT calculations like density functional theory. And the same thing can be also done with machine learning. There are a few publications already out where comparing between the energy criteria or electron volt. Then you can see here somewhere very much closer. But if it is going away, the model is going away from the applicability domain, and the structure or the chemotype is somewhere different from that of the training set, then you cannot look at this closeness. There might be a good deviation from the experimental or the actual simulation data. Another one is using neural network to predict reaction conditions. When you already have thousands of reactions and its condition, and you try to generate a model using neural network, and then when you are proposing a reaction, the model will be able to propose what could be the optimum reaction condition. For example, which catalyst, which solvent, which reagent, as well as which temperature, at what temperature. And when you're looking at deep learning applications, you're just starting with structures or smiles, and then you're trying to design new molecules, or even looking at retrosynthetic pathways. And also, when you're using the activity data, you will be able to correlate among the structural properties or descriptors along with the activity data to correlate them to come up with a model which could predict the activity for other compounds later. So this is something that we were doing earlier as a quantitative structural activity relationship, right? And these are the few schematic representations of main components of atomistic ML, where you have to thoroughly understand the data visualization part and how these interpretations could be done. Most of the time for us, when we did QSR, we would have explored about scatter plot or residual plot, or even applicability domain, we look at Williams plot. And then when you apply like categorical data, we look at ROC or confusion matrix, depending upon the categorical or continuous data. So those things also came in. So data visualization is something very important to make interpretations of the quality of the data. And this was something happened last year where they were having a competition in the Kaggle and many others, looking at different data and DNN method to see how the accuracy could be achieved, AUC as well as R square values, applying different models or methods to different data sets. Now, how does the computers or the algorithms understand the structure? This we have learned in our first class in computer science when we were going to school. Bits and binaries, zeros, ones, and zero ones, or one or one zeros. The same way, we wanted to convert our chemical structure to the bits and binaries. So they need a computer-friendly way to input the atomistic system. And this is where we look at fingerprints. So each region of the compound will represent unique fingerprints. They could be called it as a local or atomic descriptor. But overall, the molecule will be having another unique fingerprint or bits and binaries. So that's where we wanted to see how we can translate or we can make use of ML or others using other tools. I'm not going too much into coding here. Of course, you can use Python, you can use R program, or you can use Weka, W-E-K-A, Weka programs to run your machine learning models. A 9 is something that I'm using for the past almost more than 10, 12 years. I'm being using this. So I thought to introduce this, and I have also had some videos already shared how to work on this. So NIME Analytics platform is a completely open access software. It's an analytics platform where you can have workflow, automation, integration, and parallelization. The good thing is, whatever I told you, Python, R program or R package, then Weka, and many others are already integrated into NIME. And you need not to do any coding. And this is what you can download. NIME, I think, they have a latest version about 4.2. You can check that out. It works for both all the operating system, but only 64-bit. If you want 32-bit, you have to go to 9 2.3.1 versions. And then you have to set the workflows and set the memory, and then you can set the workspace. And then you have to install the extension. Since today we are talking about drug discovery, I'll specifically mention some of the extensions and node helpful for drug discovery, which will be helpful for many of the data preparation or data generation. So in order to create a workflow, first you have to go to the file menu and say create a workflow, or select a wizard for the new workflow. And then you have to just give a name. And these are the different regions of NIME. Don't feel it is complicated. Once you learn the initial part, it's so super easy. So whatever you see here on the first part, it's called servers and workflows, which are examples. And workflow coach is something that helps you or recommends you what could be the possible node could be added to your workflow. Another is something called as node repository, where it has a collection of all the nodes. Then it gives an overview, also a console about any error. And on the right hand side, something called as workflow and node description, where it talks about the how the node is being explained, how to be used, all those things. And this region is where we call it as workflow editor. That's where we play around with the connectivity. So you read a file, process it, and then you write it as an output. It's something similar to like what we write as a flow chart or algorithm at initial days when we started learning computer science in our schools. And then this is called the node repository. You have to select the right node for your type of calculations or automation that you want to do. And it also gives you the description how this can be done. Now, how do we put them into the workflow editor? Just click and drag and put them into the editor. And there are certain column filters and many more available. I'm not going deeper into it because I want to discuss about the nodes. So selected drug discovery nodes, some are paid, some are free. Whatever I have listed here, they're all free. So some requires additionally install libraries also depending upon the operating system. And most doesn't require. So it works for all the operating system also, right? So these are all nodes that is available for free. And these are the nodes that is available, of course, if you have a paid license from the respective suppliers. There are certain extensions for cheminformatics or CAD. For example, from Vernalis, you can generate fingerprints. You can also look at RMSD confirmation analysis. You can generate confirmations. You can search Europe in PubMed Central and other databases also. You can do matched molecular pairs and many more others. And when you're coming to Earlwood nodes, you can do SAR analysis like automated matched pairs, structural similarity, and virtual screening, again, ligand based. And then you can do a database similarity search, and even generating canonical or molecular properties. And RDKit is something very famous, and I'm sure many people would have heard about RDKit. And there also we have converters, 2D to 3D optimizers, ligand preparations, descriptor generations, and Marcus enumeration, and reaction mechanisms, and many other you can do there. And CDK is something very, very old. And during my earlier days, of course, we were completely dependent on CDK before RDKit was coming into picture. So we have again 2D to 3D conversions as well as descriptor, including XlogP. XlogP prediction, that's the reason mainly why we use CDK. And then we have some commercial extensions also, which is from Biosolve IT, where you can do virtual screening, molecular docking, chemical space search, pharmacophore-based search, and then you have from Chemaxon. Of course, all these these nodes are free of cost, where you can draw 2D structures, that is Marvin Sketch, to view them Marvin View, Marvin Space in 3D, and Mole Converter for the different file format conversions. But others, everything in the JChem, it's again licensed. Cresset and MOU, similarly, you have the nodes that to be, you have to pay for it. Now, let us look at the basic chemistry nodes, which is of course free and can be used by everyone. So the first part is where you can draw the structures: chemical sketcher, Marvin sketcher, and structure sketcher. Then you have file readers like SDF reader, Mol2, SDF Smiles reader. Then you have converters, Mol2 CDK, RDKit from molecule. Even OpenBabel is also available. I'm sure many of you will be knowing OpenBabel, where you can do 2D to 3D, 3D to 2D, and different file formats to other chemical file formats, and Mol converter from Chemaxon. And then you have viewers. If you want to view the 2D SDF, you can use Marvin View, Ligands Viewer in 3D, Pharmacophore Maps if you want to view Pharmacophore Viewer, Proteins Viewer, as well as Ligand and Proteins Viewer, where you want to view the complexes. Similarly, there are chemistry calculators where you want to generate descriptors. So tomorrow, if you want to apply ML-based models or approaches for your QSAR, of course, you have to generate descriptors. So this is what you call it as chemistry descriptors. You can calculate log P, molecular properties from CDK, RDKit descriptor from RDKit, molecular properties again from Indigo, and then PADL descriptors. Again, it's also generate more than thousands of descriptors. Then you have filter rate, calculate properties, again pharmacophore based, and PDB property extractor, and analyze mold D2. Mold D2, of course, works only for Windows, not for any other operating system. And once all these are generated, if you want to take an out, for example, you convert it from 2D to 3D, or some descriptors being calculated, you want to write them or save it as CSV file or Excel file, right? So those things can be also done here. These are some picked up, hand-picked nodes. So it's not that this is only about KNIME, it's more than that. And then you have the normal table readers also available. Just wanted to tell you this: S represents string, I represents integer, so that it helps you to understand what are the data types. So when you use machine learning, there are three types of data that you have to consider. One is called target. Target means if you're working on biological activity, and that is what you call it as target. And then whatever you look at descriptors, molecular descriptors, they are called as features, right? So you need to look at features and target, and before applying to it, you have to do a step, something called as feature selection. If your feature selection is not proper, your model is also not good. We'll talk about it in a moment. So these are some sample workflows that is available. I'll post the link in the chat at the end of my talk, so that you can download this, which is available in my GitHub. So you just load it to KNIME and it starts working. Only thing, you have to define the mol file in this reader or SDF file and connect it accordingly. So if you want to do a 3D optimization, you can use this, or if you want to just do a 2D to 3D converter using CDK, you can use this. Both will write SDF files. Similarly, if you want to generate environmental toxicity, if you want to predict environmental toxicity models, for example, mutagenicity, carcinogenicity, developmental toxicity, skin sensitization, fish acute toxicity. Similarly, So these are all from EPA, right? So US EPA, these models are being taken from. And if you want to generate confirmations, of course, you can use RD-Kit conformer generator, again based upon force field like universal force field or MMF. And site of metabolism, again for cytochrome P450, where you can look at the selectivity or regioselectivity of which isoform of P450. And also you can do substrate structure or substructure or similarity search between databases, or you can download the whole Campbell database, and if you want to search your molecule against Campbell, that could be also done. In this way, you are not exposing your compound to online databases. And if you want to do a pharmacophore screening, when you have an active bioactive confirmation of your reference molecule, and you can load it, and then you can do a pharmacophore screening also could be done. And also molecular property descriptor calculation, where if you want to work on SAR models. So far, whatever I have shown you is just a basic things for you to help starting doing it. uh, it doesn't look nice if I'm too much talking about advanced algorithms, and I want you to start first, right? Within 40 minutes, we cannot finish everything. So I'm just giving you some tips and tricks where you can start with generating the data, especially for drug discovery. The next one is, and this is one of my favorite, especially for ML and statistics. It's not too much weighted like 9, but yeah, you don't have too much flexibility here, but you can use many of the models. That is called Orange software. Again, you have for Windows, Mac, as well as Linux. And this is how it looks like. The nodes here, the nodes are mainly for bioinformatics, statistics, data analysis, data models, and others. Nothing too much into different applications, which I showed you earlier in 9. So here is something which I was telling you about feature ranking. For supervised problems where data instances are annotated with class labels, we would like to know which are the most informative features. The important features or the key features has to be selected for your model building. If the features does not have its own importance, then your model building also have gone to false positives, or overfitting issues will be there. So we have to take care of this missing data as well as feature selection step just before any modeling purpose. It's not only applicable for QSR, it is applicable for any machine learning model steps. Then classification tree, where we want to look at the combines interface and the visualization of classification tree with a scatter plot. When both the tree view and the scatter plot are open, selection of any node of the tree sends the related data instance to scatter plot. That's the important thing here in orange. If you make a change in the classification tree, or just a selection of the specific feature from the file, automatically it populates the other data. You need not to run it manually. So it can handle any large amount of data, keeping in mind your machine is also capable of resourceful. And this software is absolutely free. And then cross validations could be done, where I wanted to generate my data, I mean model, using logistic regression, random forest classification, and support vector machine. Then using these models, I wanted to test and score, means I will be taking the experimental data from my data that I have loaded, and I'll be taking the predicted data from these 3 different models, and then I'll be scoring with the confusion metrics to understand how far the cross validation is helpful, or which model to be selected for choosing the predictivity. Now comes the coding part, where if you are interested in Python and Python libraries, of course, we have something called as Anaconda. It's a package where it includes key softwares, Python libraries, and Python inbuilt into it. The latest Anaconda includes Python 3.8 with a Jupyter Notebook, where it is an IDE, or even you can use a Spider, which is again a IDE, which will be helpful for your coding. And there are other different libraries like SciPy, NumPy, SkyKit, Pandas, Matplotlib, PyTorch, Numba, TensorFlow, and many other, Bokeh, and many others, which will be helpful for machine learning as well as drug discovery. But I'll be listing few of the libraries which will be helpful for drug discovery. So this is the website, how it looks like for Anaconda, and you have to go to the Anaconda individual edition. and you can download any one of them. But keep in mind, you should have at least 3 to 4 GB of hard disk space available for Anaconda, because for the download it might be showing you only 500 MB, but once you load a lot of libraries, of course you require tremendous amount of hard disk space available. So this is how the navigator looks like. And the navigator, you can load the Jupyter Lab or Jupyter Notebook, but in that you can do the coding, or you can load Spider, where you can do the IDEs. So I'm not talking about a Python or anything as such right now, because I don't want to scare you with the coding or scripting. But let me tell you which are the key important libraries which you have to consider when you're working on drug discovery, especially when combining with machine learning or other data analysis. DeepChem. It is an open source tool chain for deep learning in the drug discovery, quantum chemistry, and other life sciences. RDKit, which I have already mentioned, which is available for NIME also. It has some functionalities include reading and writing molecules, substrate structure search, chemical transformations, and molecular similarity. And we have something called as Cactus, which is a universal scriptable chem informatics toolkit with a large collection of modules and property computation. And Symfony, it's a chem informatics toolkit which includes OpenBabel, RDKit, and CDKit, and Indigo. And similarly, we have MD analysis and MD Traj, which will be helpful for analyzing the trajectories of molecular dynamics. And NGL View is a 3D visualizer which is available within the Jupyter Notebook for visualizing your trajectories or complexes or interactions. NumPy, Matplotlib, Scikit, these are, or Seaborn, these are all related to data analysis, machine learning, statistics, multi-dimension arrays, matrices, and Scikit especially for classification, regression, clustering algorithms. GROMACS wrapper, whoever is interested in GROMACS molecular dynamics, so they have a Python wrapper called as GROMACS wrapper. So the Jupyter Notebook for GROMACS also I'll be sharing just after the talk. So if many of you are having doubts how to install these codes and all those things, I have already generated a demo video which will help you how to install each of these steps, including Amber tools, OpenMM, GROMACS, and many other tools which will be helpful for drug discovery. And all the installation links are also available in the GitHub. Under the basics, under the science, you will be having the links which is useful for the libraries which are listed out will be also there, how you, or what is the command that you have to give in order to install this in the Anaconda. So that's all from me today. I think I kept the time. Just before I finish, when you're looking at machine learning codes or Python codes, you can see many codes are available. But in that way, you're not going to learn anything, just copy pasting and doing it. How I was able to learn was, of course, I looked at many codes, tried to understand it, implement it on my style, and then wrote the code myself. In this way, when you apply with your own ideas and creativity, it might be much better than the others, or might be bad, but still you're trying your best to how to adapt it from other codes. And similarly, for all the students, until you try yourself and train others, you cannot be an expert. Just learning, listening doesn't make you an expert. You have to apply somewhere, make mistakes, try to overcome that mistakes by finding out how it to be corrected, and that's where you become an expert. So thank you so much. I just gave you an overview about the drug discovery and drug candidate selection using ML, using the tools like NIME, Orange, and Python libraries. But if you have any questions, I'm happy to take that. Thank you so much.
Thank you so much, sir, for such an informative session, and especially for the tools that you shared. So, first question, there are many questions in the comment box, but I would like to open the Q&A session. That I am an undergraduate student of B Pharmacy, and I'm very much interested in the artificial and machine learning. Recently, I did a course on artificial intelligence, but it was so difficult to understand that I couldn't move on to machine learning. So, is it
Mhm.
necessary to learn Python in especially drug discovery so that I can understand the papers written by and use it for my own use?
A very good question. See, for machine learning, the first and foremost part that you want to learn is statistics. Many of them think that you don't require statistics for machine learning. Without understanding statistics, don't ever think about machine learning. Number one. Number two, you also need to understand how the data interpretations could be analyzed. So you got a scatter plot or you got a confusion matrix. What that number is going to represent? If you're not, or if you're not knowing how to interpret them, then what is the use of doing machine learning? For me, these two are the important part. Now, the other important part comes if you want to become a developer. If you want to become a developer, you need to know a good amount of mathematics, especially probability, linear algebra. I know people now get scared now when we talk about mathematics and algebra, right? And then you should know about Python. It's not about only about Python, which library, which parameterization or model tuning to be used. This comes from experience. So that's why I said copy pasting is not going to help you. Try and work around with different tuning parameters and then understand. So this will help you. Now, if you think that I don't know Python, I hate coding, don't worry. Then you use Nime or Orange. You don't need to do a single coding there to generate ML models. So it's your choice where you want to be positioned.
Okay, thank you. That was a very nice explanation, sir. This question is also related to that, but I can see one question here. It's called "Is Nine free?" The tool you shared.
Yes, the analytics platform is completely free.
So, moving on to next question. Parvesh Sharma is asking, how can we differentiate supervised and unsupervised machine learning in simple words?
Huh. Uh, I think I have to share my slide again for that for that explaining with some examples. But I think that we have to put it across on looking at supervised and unsupervised. It also depends upon the kind of data that you have and also what you wanted to solve or what is your objective. So it again takes at least 10-15 minutes to get a thorough understanding. So I will post the link in this particular video. I'll share it with organizers. You can go through that for a better understanding. Yeah.
Okay. So, moving on to next question. Please explain about feature selection in QSAR.
So feature selection in QSAR, sometimes mostly we have used with genetic algorithm, right? Using genetic algorithm, you can do the feature selection. But the problem with genetic algorithm, you know that as when we iterate, we don't get the same thing, right? And also we have to look at the intercorrelation, like how far the descriptor with one descriptor and the other descriptor, how far they are being correlated. If they're too much correlated, for example, more than 85% or 90%, and if it is coming within the model, we still have to consider p-value and other its significance. But when you're talking about a biological activity of using in QSAR, the feature selection also should have physical meaning and significance. What particular feature means descriptor that you have chosen is having some kind of relationship or physical meaning against the activity that you have chosen. That's where I recommend you to read. There are plenty of blogs available for this particular terminology: causation versus correlation. You can correlate with anything, but until you know the causation, it doesn't make any sense. So when you go through that articles where they talk about causation versus correlation, that's where you understand the importance of feature selection in a statistical perspective as well as on a physical significance perspective.
So, next question is, most of the students in India who choose healthcare are weak in mathematics. Can you please share how can we make our future in machine learning in healthcare perspective?
That's what I heard recently because my wife is also into data science. So she was telling me that people have to decide who they want to be: data science scientist, data analyst, or a data science engineer. These three categories are there. So when you look at the healthcare, you very rightly said when people think that mathematics is difficult or weak, that's where they look into healthcare. So in your healthcare, I'm sure you need not to break your head to develop something. Rather, you will be mostly looking at developing a model, or you wanted to analyze your data. So in that perspective, I don't think you need to know too much of mathematics, but you need to know statistics and interpretation. So there are tools, something similar to, for example, Power BI or Tableau or Spotfire. Of course, they are all paid. But when you alternates, if you look at, you have Nine and Orange. If you are able to analyze the data and interpret then I don't think this will be a great challenge for the healthcare sector. So still you will be able to analyze the data.
So next question is whether it's required to validate the data generated from the machine learning program.
Absolutely. Until unless you validate, for example, when you do QSR, you have training set and test set. Now, where is the validation set? Right? Now many of them told me, sir, because the software did not give us an option to generate a validation set. Come on, once your training set and test set is done, there should be a validation set to validate whatever the models you have generated can reproduce the model or is a reliable method. So certain methods have different statistical parameters to validate. One is standard deviation, one is accuracy, one is confusion metrics, and many more others. So we have to 100% validate the data before deploying the machine learning. For machine learning has three different steps. One is generating the model. Next is testing or validating the model. And the third is deploying the model. So before deploying, if you have not validated, tomorrow if you give some data that is going to be predicted, it's going to be wrong. So you have to validate it. So validation is a very much required.
Moving on to next question. So could you please let me know what should we do to get into a good control in CAD and chemoinformatics part?
That's where we have to understand one thing. My master's was on bioinformatics. Don't think drug discovery or cheminformatics is completely bioinformatics. No, they have been positioned in each expertise. When you come to cheminformatics, you need to know a lot more about chemistry, structures, fingerprints, and very important, database management and statistics. Very much. That's where we call it as chemometrics. I don't know how many of you are aware about that terminology, chemometrics. It's in under cheminformatics. And you need to know about data management, for example, like SQL querying, Oracle, or something like that, right? So that's where cheminformatics part come into the picture. And when you come to CAD, initial part of target identification, validation, it's all good. We talk about proteomics, genomics, systems biology, or network pharmacology. But then we enter into docking and pharmacophore optimization. It's all about chemistry. It's all about organic chemistry. So don't try to overdo everything completely by one person. No, hand in hand, you have to collaborate with the experts. So each person has to look into each area and domain. So when you want to have a good control, first point is try to understand where is your expertise and where you yourself to be positioned in the great large phases of computer aided drug discovery as well as in cheminformatics. That's the first thing that you have to understand. Understand what you're good at it, and then try to see where you fit into it. And rather than trying to do everything. Now, many of the people don't do molecular dynamics just because it is too much of biophysics, too much of mathematics, too much of structural crystallography, and many other things. But it is still easy if you understand biophysics much more. So that's where we have to position ourselves. Yeah.
Okay. So I think there is last question. Yeah. We'll move on to next question. This is Sir has covered already this. Can you
Yeah.
please suggest some internship or job opportunities for students interested in drug discovery using machine learning?
Yes, I could see, Ishika. It's about internship or job opportunity. First of all, when I go for many of this entrepreneurship summits, I hear from students, until unless a company or some organization give us an opportunity, How do we achieve experience? That is the biggest concern all the freshers used to ask a question. But then I said the same student that you had lot of opportunities during your internship to get that experience or during your project. It is not necessary that internship should be done in a company. See, certain internships, I don't want to comment here. It's not the actual thing that you wanted to learn. You're doing something else to earn some money or pocket money. I don't want to comment here. But when you try to go for internships, choose the right one which is going to help you for your future career, whether you want to stick on to the same domain that you're learning or you want to switch on from the domain that you're working on. It's your choice. So choice of internship should be your choice of your passion and what is that you're looking for the next 5 years. That is one. Second, when you're looking for internships or any other, have a good passion of reading. Until unless you read and learn and on a self-paced learning, this is not going to help you for aiming or achieving experience during your academic career. So this is very important. Now, I know the answer for this question is just telling you some companies or some, that doesn't help you, because tomorrow if I'm going to tell you, please go to Syngene, please go to Dr. Reddy's, please go to Cipla, they won't simply take you. They wanted to know what you know by yourself. What is that you have self learned yourself? What is the experience that you know from your academic career? It's not about that you got this much CGPA from GPA from this particular paper. No, what is the experience that you achieved from a project or academic project? So first try to achieve that, and second, try to take a lot of courses which is available for free. Even in Coursera you can audit the courses on statistics as well as on machine learning, including Python. So try to upskill yourself. Since last lockdown, I am sure that many of the students got opportunity and more time, even myself, to learn many things sitting at home. So upskill yourself, and then you will be a ready, experienced person for the fresher to apply as a fresher. Yeah, that's my take.
That was Yes, sir. That was a very apt answer. So thank you for the session, sir. And I would like to hand over the stage to Paresh, sir, now.
Thank you so much.
Thank you, Dr. Pillai. It was very nice presentation and very well explained. And for me particularly, to know about some basics about machine learning in drug discovery, it is very new to me, because today, we have combination of a researcher and entrepreneur as well. So first we heard, we listened to the Dr. Pillai. So he talked about the very latest improvement in combination of machine learning and drug discovery, that how these two different discipline can combine, and interdisciplinary work can evolve for making something very new one. So with this one we have our friend Dr. Andrew Tait from Canada. So he has done the research, and he is also an entrepreneur. So this is a different combination, because we learn first about the machine learning drug discovery, and now we have Dr. Andrew Tate. So in reality, he work on the research, and then he developed the products, and then he's marketing his products through his laboratory, that Tate laboratory. So we have different flavor for today's webinars. So I would like to introduce uh, Dr. Andrew Tate, because he is coming from the Canada. So he has done his BS and MS from the Concordia University, from all the way from East Coast in Canada, from Quebec and Montreal. He completed his master and BS degree, and then he moved totally to the west coast, to the Vancouver, British Columbia, where pursued his PhD in University of British Columbia at the chemistry department. So where I got a chance to work with him for 1.5 years. So we work in the one of the team together for 1.5 years, and we produce some very uh, important research also, and we have also shared, many good moments together in a laboratory also. So I recall, that working with Andrew, and we used to go for coffee at the student union building at the University of British Columbia. That every day, almost we used to go for coffee. And today here we are, after 15, 16 years, I'm introducing him for this particular webinar because he completed his PhD from UBC in 2011. And then after completion of his PhD, he also worked at the biotechnology network at the UBC. So there he was a director of this student biotechnology network for around 1.5 years. And then he also, inspired the next generation innovative leaders in biotechnology and biopharmaceutical studies. And after that, in 2011, he also founded his Tait Laboratories. So Tait Laboratories, he is now currently president and director of this laboratory. And his main purpose, means main mission of the Tait Laboratory, is that making tomorrow's medicine from today's food waste. So this is his mission of the company and the product. So I I would not take more time to explain about his products and his research experience, but we can hear directly from Dr. Andrew, my friend Andrew, and we can see that how his journey from researcher to the entrepreneurship, how it went, and how he took his product to the market from the laboratory. So it's going to be a very wonderful experience for the listener and the viewer, that how one can take their product to the market through their research. So here, my friend, welcome Dr. Andrew Tait for accepting our invitation and delivering today's talk. Dr. Andrew, floor is yours now.
Well, thank you very much, Dr. Paresh Dave. Paresh, it was good to see you, and in the spirit that we once shared, I'm having some coffee. So good morning, everybody from Vancouver, BC. I know it's evening where you are, but it is just past 6:30 in the AM here. So I am awake, thanks to a little bit of caffeine.
Thank you for waking up.
Yes,
to
well,
this
my pleasure.
morning. So.
My pleasure.
Yes,
yeah.
yes.
So anytime I get the chance to speak about my experiences from research and academics, all the way through to research in industry, to inspire students that they should follow their passions, to keep asking questions, to stay curious, I think I jump at that chance. So I want to thank Dr. Pillai for his interesting talk first and foremost. And one of the key messages that I learned was, or wanted to also share, was make mistakes, right? If you stay curious, you keep exploring new things, then you're bound to make mistakes, and that's okay. And this sounds like it might be a little bit of a trite bit of advice, but I think oftentimes when we're in an academic path where we're just studying and trying to make good grades, that we forget the importance of just remaining curious to the topics that we're learning at hand. And I was very interested to learn more about Dr. Pillai's mission in machine learning and chemistry. I did have a background in molecular dynamics and simulations as part of my training, learning a little bit of Python. I, on the other hand, was also not good at math. I did see some failures, but picked myself up and brushed myself off to continue on learning more about the things that made me curious. So many of you here today are curious in the very broad subject of life sciences. And so without further ado, I'll tell you a little bit about my background, because I've always sort of had a passion for biology, for chemistry. And let's just see, I'll start sharing my screen here. If you just bear with me a moment. Okay. I believe that you should all see my screen now. Okay, well, this pretty much defines what we think of as a scientific discovery, a potential drug candidate that we discover in the lab. It's
Andrew?
actually, yes. Can you hear me?
Yeah. Can you, because I am not able to see your screen yet. So
Okay, let me try that again. Let's see.
yeah, there you go.
Thank you so much, Presh.
Yes.
So as I was saying, this pretty much exemplifies what drug discovery is, where you find something exciting in the lab, a potential drug candidate, but you still have to go through the years and the millions of dollars that come along with an approval by a government regulatory body. And as we know, that most of the world's drugs are developed first and foremost for the US market. There's many reasons for this, many of them financial, but yes, I think it does let the air out of the balloon, so to speak, when you think about all the other things that most of us scientists don't really like, the commercialization aspects, the regulatory hurdles. So there is a lot more behind the scenes behind just a simple discovery in the lab that we all get excited about. One of my early mentors said this to me, and maybe I'm paraphrasing here, but science is hard, the business is easy. So if you're in school right now, just focus on the very interesting aspects of science, and do try to combine that with other areas of curiosity. In my case, I did study hard in chemistry, but I would also attend seminars on entrepreneurship, getting in touch with people in the industry. So not just carousing with academics, but meeting people who were executives in biotech and pharmaceutical companies. And now these days, it's even easier more than ever. There's so many different social media platforms. There's the advent of Clubhouse, where people can just get on and speak to each other in real time. And conferences like this kind of break down barriers in uncertain times, like during the pandemic.
Wow.
A lot of us also feel kind of alone. And so basically, this is some words that I live by, that, focus on the science, but also remember that, you can also build a team that help you with the business. So I won't belabor the intro that Paresh had so generously given me, but just want to say that school wasn't always easy. I was a top student in high school, and I think a lot of you do, perhaps struggle with some courses that you otherwise would find interesting, but the grades just don't present themselves. So I did have some struggles early on in CEGEP, which is right after high school, but I really found a passion for biology and chemistry. I'm not so much on the calculus, which maybe some of you can relate to, but I did manage to finish my bachelor's in biochemistry, having done a research project on an enzyme called enolase involved in basic glycolysis. And that really sparked my interest for laboratory research. And I would suggest that you all give that a try. I mean, you don't have to be working in a lab per se, but really, in the message that Dr. Pillai was sharing is, make mistakes, like try things out for yourself. Don't just read about them. Although reading is very important, but it's really a matter of putting yourself in these situations that make you uncomfortable, right? With new learning new things and expanding your horizons. So I jumped into a master's in chemistry in the lab of Dr. Pulowski. And there I was learning more environmental biochemistry. So studying organisms and enzymes that break down toxic chemicals in the environment, like chlorinated biphenyls, and trying to design enzymes that would break down a wider range of these toxic substrates. So then shifted gears and also shifted locations, moving to Vancouver, BC, where I did a PhD in the lab of Dr. Susanna Strauss, learning more about membrane protein biophysics using solid state NMR, where I brought a background of protein expression and purification. So it was an interesting marriage of the theoretical and experimental worlds, but again, very interesting. And I learned a lot over that time, also from Dr. Dave. Having him in the lab when I started was a wonderful asset to learn so much from his rich expertise. So fast forward another few years, about 6 years later, near the end of my PhD, I founded a company. So how did I do that? Well, I'll tell you right now. It was a big transition, let me tell you. So to go from a PhD student to entrepreneur, you can see in 2008, I was keeping a different company than I did in 2016. So it's not a very straightforward path. Success seems like it's a straightforward path, but there's the expression, the 20-year overnight success, because that path to success always kind of meanders in different directions and taking you down different alleyways. And in the words of my master's supervisor, science is about that, especially, is going down a lot of alleyways and figuring out which ones of those are blind alleyways. So turning yourself around, getting out of that dead end alleyway and going down a new path. So this is completely shifting gears now again, talking a little bit about what inspired me to start the company. And it really was my PhD research. At the beginning of that degree, I couldn't have guessed that I'd wind up with starting a company. So multiple sclerosis is a disease that I was studying. And if you know a little bit about this disorder, it's where the myelin sheath, which is the insulation on nerve fibers, breaks down, leading to dysfunction. So once that nerve fiber is exposed, once the myelin breaks down, then you have breakage. And it's kind of like the insulation on a wire breaking down. And so when you have a lot of that happening, you have plaques showing up in the brain, leading to a life of disability. So difficulty moving around, a lot of pain and discomfort. Now, this kind of sums up my 6 years of research in the lab, where we had hypothesized this generalized model where a viral protein from human herpes virus type 6 that I was studying, called U24, could potentially compete with other very important enzymes located within myelin, that by competing with these enzymes, you would potentially see the dysfunction, the breakdown of the myelin here. So some of the experimental results we had, on the left here, you see there's a competition assay for phosphorylation with U24 versus myelin basic protein, which is a very important structural protein in myelin. And on the other side, you see that we showed that you could compete with binding to Fin tyrosine kinase in the SH3 domain. So this is an SDS polyacrylamide gel. And basically, the two important lanes are in lane 4, you are able to pull down the U24 protein. But when you remove that very important N-terminal domain, then you're no longer seeing that protein pulled down, which would be lane 8. So we published these results in peer-reviewed journals. And yeah, so this was a diagram that I always referred to. So you never know where something that sparks your curiosity is going to show up. I just would throw this around in academic presentations saying, well, the protein I was studying, U24, could interact with this enzyme pathway in myelin up top and down below, these two different pathways. So competing with myelin basic protein for phosphorylation, and then Fin tyrosine kinase at the bottom there. And also in this paper, they mentioned that there was an herbal medicine 18 herbs and spices that could reduce or essentially reverse demyelination in animal models. So that got me very excited. I began to talk to people about this, multiple sclerosis clinicians and academic scientists, because the data was very compelling. You could see that not only did it repair the myelin in MS model mice, but also older mice. So you could see a lot of applications here. So on the left, you see that the number of myelin fibers goes up when they're fed this herbal formula. And on the right, you see that the G ratio goes down, which means a greater thickness of the myelin as they're in these mouse mice that are fed this herbal formula. So the same thing goes for the mice fed cuprazone, which is the MS model. That basically you do have the thickening of a myelin and also the number of myelinated fibers going up. And when you create knockout mice, mutants, that you abolish that remyelination activity. So actually two MD, PhD local biotech execs were telling me that the only thing that you can do now is go back to the lab and identify the lead candidate in that mixture of herbs. And that's the only way to go. But that didn't really sit right. I felt that we could potentially get some anecdotal evidence, maybe screen the raw herbs themselves before actually trying to invest time and effort to screen all of the different molecules against many different assays. So this is where I had the opportunity to work with a naturopathic clinic local here in Vancouver, headed up by Sonia Jensen. And so they imported this formula. There's equivalents found in traditional Chinese medicine and traditional Japanese medicine. This one's called Ren Shen Yang Ying Tang. So you can see all the different components. And as you can appreciate from a pharmacological point of view, many different alkaloids, bioflavonoids, what have you, that could have a pharmacological effect. But after some follow-on research, it was shown that it was actually citrus peel that could stimulate the remyelination in animal models. So this kind of is a formulated model of what's happening when the animals are subjected to oral administration of mandarin orange peel. Is that you have, first of all, demyelination and an accumulation of oligodendrocyte precursor cells that build up, and administrating the mixture where hesperidin and nerirutin are two structurally related bioflavonoids. They could actually stimulate the enzyme pathway I was studying. That if you recall, I said U24 protein from the herpes virus could potentially inhibit. So compounds in citrus peel could essentially reverse that, causing maturation of the oligodendrocytes and then remyelination of the nerve fibers. So very, very exciting stuff, but needing to be substantiated beyond animal models. But in the meantime, I said, why can't we just import high quality mandarin orange peel, a waste product from the juicing and canning industry? Found out that 4 million metric tons of mandarin orange peel go to waste each year as a result of cast-offs from the juicing and canning industry. So I figured, because there are 80,000 people in Canada that suffer from multiple sclerosis, maybe we can try to see how this works anecdotally in the hands of clinicians, to see how it might affect their well-being. So I had the idea that we could do that, and also at the same time, in parallel, screen for active compounds and develop a drug candidate that we could then potentially take to market. So I very excitedly and perhaps stupidly bought some equipment with all the money that I had saved up. This is equipment found on auction from another company that had closed in my hometown of Montreal. So I shipped out a centrifuge, a freeze dryer, a pump to go with that freeze dryer, and some hot plates. And I don't recommend this. I also don't recommend renting real estate to start a lab, but that's what I did. Basically, after founding the company in 2011 and moving into this old shipping office, we had to close down shortly thereafter. But that's not bad news. What I had figured out we could do is we can outsource some of the development process with the help of government grants. So we didn't have to actually do the work ourselves, but we could design the experiment so we can figure out how to make some novel extraction methodology, clean extraction methods, green chemistry as well. So after closing our doors, we managed to partner with the government and with other existing extraction facilities. And so we basically turned the remaining 4,500 Canadian dollars that I had saved into $128,000 by getting these government grants and loans from these following organizations. So very blessed, very lucky to be able to sort of multiply that by a factor of more than 30 to enable a greater level of research to be done and development. So this is myself and my early partner, another one of India's terrific sons, Dr. Harish Vasudevan. He was a co-inventor on our extraction technology, and this is us in our entrepreneurship at UBC office. We managed to land some office space there in the incubator, and we're very happy with grins on our face because this was our very first paycheck. So we developed our own extraction methods, and rather than take the long and lengthy route to market market something for multiple sclerosis, we saw that there was a nearer term ability to market a product for digestion. Right? Mandarin orange peel is part of traditional Chinese medicine, and it's been used to reduce gas, nausea, bloating, diarrhea for hundreds of years. So it's a very valuable traditional Chinese medicine where they believe that the peel gets better as it ages. And the science behind that is that when the peel ages, the matrix tends to break down. So the cellulose, the pectin, so sort of a dry aging fermentation, making it easier for when you heat the peel in hot water, as the Chinese would do, and drinking that solution, it makes it easier to extract the polyphenolics and the bioflavonoids and maybe short chain pectic fragments that can have a diverse array of biological effect, from helping the growth of good gut microbes, so improving the diversity of your gut microbiome, all the way to working directly on the gut lining itself, so repairing the gut epithelial layer, and of course reducing inflammation, right? Which is all the rage these days to talk of dietary components that can reduce inflammation and the disease that results. So inspired by the early beginnings of where we started for multiple sclerosis, we have the name MS Plus, as in Mandarin Skin Plus. So most of our customers that are continue to buy our product to this day buy it for digestion, and we now know that in digestion it underlies so many different autoimmune diseases, potentially including multiple sclerosis. So we don't actually have to market something to have what we think is a very positive change on the world. We market it just for digestion, and we still continue the research to this day to support that gut-brain connection, if you will. So healthy gut, healthy brain, healthy other organ systems of the body as well. So inspired by the way the Chinese processed peel, we developed our clean water extraction technology and filed some patents on that. So coming from a chemistry background, it was relatively easy to convert data and reports to a patent application. But again, we needed help. We needed people who were experienced, and luckily we had that resource at the University of British Columbia. This underlying theme I'm hoping to achieve with you is that it's very important to continue to reach out to people when you don't understand or need a little bit of help in moving things forward, because you can't do it all yourself. There's the expression, it's not how, it's who. So it's about surrounding yourself with good people. And it's so easy to reach out on LinkedIn or via email or any other social media platform to really understand who can help you and answer those questions that you have burning as a result of your curiosity. So we moved offices. So we then went to Discovery Parks. We won some kind of business planning writing challenge, and then went to the top floor of Granville Square. We subletted a small space overlooking the beautiful bay and the mountains here. And that was just housing with a biotech company, because I got to know the owner and one of his executives, and they said, sure, we've got a few extra desks. So we moved in there, and also went mainstream with our product rather than just selling to a few registered dietitians and holistic nutritionists. We launched a product that went on shelf at pharmacies across Canada and also via e-commerce. So I had no idea how to do this when I first started, but began to build steadily a team of people that could be helpful to bring the product to more people, to improve their digestion, and as I alluded to, perhaps other conditions as well. I thought this was interesting, and maybe you've heard of something called creating a vision board. I had taken this photo of the inside of a pharmacy, and I would show people where we thought our product was going to be on shelf. And we thought, here's Procter & Gamble's Align. That's probably one of the best-selling, hottest products in the pharmacy for digestive health. We want it to be right up there next to them. This is at a trade show a couple years later. And there you go. We're not right close to them, but this is actually, very similar, a pharmacy looking shelf, and we're up there on shelf next to very popular brands like Metamucil, Pepto-Bismol, Gas-X, and there is a line you'd see right down there on the end. So not far off, but I'd say that's very powerful when you can manifest your vision and show people, and other people want to help you get there. And it turns out digestive health has become a massive market. We didn't think that this is where we'd end up, but in Canada, there's 20 million people suffering from digestive disorders, and our customer set, it turns out, primarily women, where irritable bowel syndrome and inflammatory bowel disease are more frequent in them, and also other digestive diseases as well. And you're familiar with this funny little emoji, the poop emoji. And it turns out that while it's taboo to speak of your bowel movements, this is really something that is expanding very rapidly. It is helping people to understand what a good bowel movement looks like. What's interesting is that we've packaged the Bristol Stools chart along with our product, and customers love it. I think there's a lack of education on what a good bowel movement should look like. You want a type 3 or type 4 here. Parents also use this Bristol Stool Scale chart to educate their children. And on the subject of AI and machine learning, we are partnered with an app company so that people can take photos of their bowel movement, sending it to gastroenterologists, so that you can get a much more accurate and precise representation of your bowel movement. So if you're doing a dietary intervention, you actually get to see what is working for you to improve your bowel movement. And then, as I said, improving your bowel movements could potentially impact positively other parts of your life as well. So the research continues by talking to more people, networking. We managed to secure more research and development money to continue to work with the University of British Columbia in the Faculty of Land and Food Systems, also more NSERC money to basically, now that we have that brown powder that we sell in market, what does it do? Like, what is it that's unique about what we've made compared to anything out there? Well, this is just a preprint I had from our team that we published. So we looked at the anti-inflammatory capabilities, looking at certain compounds that are in there that contribute to that anti-inflammatory activity, namely nobiletin and tangeratin, which are two structurally related polymethoxylated flavones. And I don't think you can see this too well, but basically, if you look at this paper, you can see that we have a leaky gut on the left, where there's an increased amount of intestinal permeability, and in the presence of our extract, it's essentially repaired. That you could reverse that leakiness or hyperpermeability to have it more healthy gut status, and presumably improve the gut flora as well. But we're very careful to not say leaky gut syndrome. Leaky gut syndrome seemed to be a pseudoscientific term made up by early supplement companies to sell more supplements without actually showing that their product did anything. But we know that increased intestinal permeability is a real thing that experts around the world are beginning to embrace as underlying a large number of diseases that we wouldn't traditionally think of as being digestive related, like neurological ones, including multiple sclerosis, but also Parkinson's and Alzheimer's as well. So what does it take to be an entrepreneur? I mean, remaining curious is one thing, but actually staying ahead of the curve, creating goods and services that people want. Everybody's heard of Netflix, but there's also the downfall of Blockbuster, once in its heyday being the biggest video rental store chain in the world. Sadly, I believe is only down to one store after being at something like, was it 5,000 stores or more in the US alone. So it's really understanding. And in our case, when we started the business, talking about upcycling food waste to medicine was not something that was sexy or something that people wanted to talk about. But now when there's more interest, or I would say concern with climate change and moving more to a plant-based diet away from consuming animal protein, to having plant-based protein alternatives, and that effect that it has not only on the planet, but on human health, that's something that people want. So we're seeing that we're changing the conversation, describing what we do as really something that people can buy into. And it's not just, oh, there's a lot of mandarin orange peel lying around, let's see if there's something useful that we can do with it. We've been overlooking a lot of things. Things that we see as garbage or as waste, but there's not only nutrient value trapped in this waste, but medicinal value, things that can actually turn the dial on disease, things that can be developed into clinic foods, which is a regulatory path in the USA. So while one of our advisors on our team has worked in the botanical drug review team at the FDA, so understanding what it takes to bring plant-based molecules to market as drugs, there is this burgeoning pathway called medical foods that we believe we could potentially be the very first food waste that could be validated as a medical food for a digestive disease. So yeah, there's a lot of people that are involved in Tait Labs, and a lot of people that are associated, so not full-time, just part-time helpers or employees that can help with any number of these things. And these people exist around the world, other side of Canada. Some of them are in India, some of them are in the US, some of them are in Peru. So you just got to keep talking to people, stay curious, and I hope you could tell from the sound of my voice that, despite it being very early Sunday morning here, I am absolutely curious and revved up to get out of bed every single morning to talk to new people and to share this passion. And it's contagious. So if you continue to stay curious, keep reading, keep sharing with people, then yeah, anything is possible. And I don't mean to sound trite, but yes, it's true. I mean, just keep sharing your ideas, and you never know what you can do to change the world. Family is important, right? The love of family, that means everything. And as Per said, I moved from Montreal to the other side of Canada, to Vancouver, BC. And a bit of news is that my wife and I are actually moving back to be closer to our aging family, which means that there's more new opportunities that await for us there as we move back to our home area of Montreal. But surround yourself, family first. Just remember that. So we've had a lot of success going across Canada, looking for partners to enter other parts of the world. As said, we've been in business 10 years, and there's still so much to accomplish with more clinical research. We expect that we can put our novel ingredient into other people's food and beverage, and also get into other markets as well. And yeah, we felt it was important to see how people enjoyed the product. So we did some trialing as well. So very far away from the research, but this is consumer research. And we maintain an average of 4 on 5 stars. So very interesting to see how people have used the product to alleviate their digestive symptoms. And I think on that note, I'll just say continue to network, right? I cannot stress this enough. Is just keep talking to people, keep learning, share your ideas. If you have an idea about something, don't just keep it to yourself. I think there, as an entrepreneur, the tendency is to try to keep things in stealth mode. There are certain reasons for that at certain times, but by and large, I'd say that in the meantime, you should be talking to more people if you've got even what seems like a crazy idea that you may be coming up with during your studies. Just try to validate that by bouncing it off other people. So yes, I mean, as I said, networking is important. If you want to reach out to me, the best way is, well, on social media. My handle on Twitter is Taitlog, LinkedIn, but also here's my email address, info@taitlabs.com. So I'm going to turn control back over to Dr. Dave here.
Yes.
And
So,
if there's any questions.
Very nice presentation, Dr. Andrew. Because, frankly, to speak you as a Dr. Andrew, it's proud and also, some kind of different feeling that to say, okay, Dr. Andrew. But I would like to start, by your last slide. You said networking is more important, because I also started working by myself from last 5 years, and then that is the key message I understand, and I am also going through that. Networking is the most important part for any business. Even if you are a startup or if you are doing any business, even if you are a student and you wanted to build your career. So for the student also to build up career and to make connections with the different faculties or different entrepreneurs in the field, it is more important to build the networking. And nowadays LinkedIn is also available, so networking is the main important and main message for this one. So having
Absolutely.
said that, yeah, I have one question, for the medical food term you used. So whether do you require, any kind of regulatory approval for this one or not?
Yeah, well, the term is clearance, right? So unlike a drug, there's a clearance process that the FDA basically has no objections to your claim to medical food. So you can have a medical food for any number of diseases, right? And some of the ones that are on market now are for irritable bowel syndrome. There's another one for Alzheimer's, and basically you're meeting a nutritional need that is not met through diet alone. So you have a specially formulated composition. So in our case, it might be a hyper concentrated citrus peel extract. And so you do similar clinical trials, right? So almost like you go to a phase 3 clinical trial to validate that it's both safe and effective. But you don't submit anything really to the FDA. You basically have to have that in case they ask for it, that data that supports that it's specially formulated, but also safe and effective for the disease you're making. So you could say this is a medical food for irritable bowel syndrome or multiple sclerosis or what have you, but you had better have that data to say that it's safe and effective.
Okay. So once again, thank you, Dr. Andrew, for joining with us and accepting our invitation and to spend, your Sunday morning with us. So thank you once again for joining today. So
Thanks, Dr.
now,
Dave.
yeah. So now this mic over to Valika.
Yes, sir.
Yeah, please take care.
Thank you.
Yes, sir. So participants still having any queries regarding pharmacist and also the sessions, you can post in the chat box. We can answer the questions and even the speakers regarding the sessions. So moving on to the further session, I would
The
like to
Pharmacist 21, presented by Operant Pharmacy Federation, Corporate Platinum Partner Integro Pharma Limited, Bangladesh, in association with IP Movement, GPAT Discussion Center, Pharma Tutor, Placement Partner, Atish Management and Consultant, Academic Partner, Chitkara University, Dr. D.Y. Patil University, RK University, G.H. Rai Soni University, Vishnu Institute of Pharmaceutical Education and Research, Parul University, KB Institute of Pharmaceutical Education and Research, Pramukh Swami Science and H.D. Patel Arts College, Sri Kokila Ben Karsan Bhai Patel Girls Science College, Sri Ramakrishna Institute of Applied Science, Reams College of Pharmacy, VV Institute of Pharmaceutical Sciences, University of Sciences and Technology, KGR Institute of Technology and Management, Farooqia College of Pharmacy.
Yes, moving on to the further session, I'd like to introduce our next speaker of today's webinar series. Our next speaker is Mr. B. Venkteshwarulu, Assistant Drug Inspector, Central Drug Standard Control Organization, Director General of Health Services, Ministry of Health and Family Welfare, Government of India, West Zone, Mumbai. Sir will be speaking on the topic pharmaceutical entrepreneurship, opportunities and challenges. Over to you, sir. Sir, you're on mute, sir. Kindly unmute your mic. Sir, still you are not audible.
Hello.
Yes, sir, you're audible now, sir. You're audible now.
Hi all. So congratulations on you and welcome to India 20 wonderful presentation regarding drug discovery and all. So that's right. So myself, Venkteshwarulu. So I'm doing assistant drug inspector with DCGH, the Federal General of India. Just I'm going to quick highlight about one of the important opportunities. So right now available in India. So we see all
Venkteshwarulu
like India is
Galu,
considered India
your
is considered
volume is little down.
hello.
Sir, kindly raise your volume up, sir. You're not, yes, you're audible, but your volume is little bit slow. Your voice is low, sir.
Hello.
Yes, sir,
Little
it's
higher.
okay now.
Is it okay? Is it okay? Is it okay?
Yes, sir, yes, yes.
So India is considered pharmacy bowl of the world, and so in the current situation, so we are doing like we are manufacturing sector are we are number one to vaccines and the parenterals and number one palmitate on electronics. So we are exporting to all the countries across the globe. So when it comes to here, so my presentation today is going to focus on practicing pharmacy in Latina and the opportunity for the pharmacy graduates or post-graduate graduates interested to be pharmacy practicing agents. So here, even though we consider the pharmacy bowl of the world, so there are a lot of opportunities to start or to the practicing pharmacy is very poor in India, apart from when we compare with the developing countries and Western countries. So here, mostly two-tier cities, one-tier cities are there. So, but when it comes to distribution of the medicine to the last mile, last end point, it is very poor. And here you can find out a lot of educational opportunities. For example, I will take you so wellness forever. So one of the three Gujarati pharmacists they have started with pharmacy distribution. So and it is taken up by Serum Institute of India. So they are going for like IPO and many examples are there like Medmade is there and one MG is there. Medmade is taken over by Reliance Life Sciences. So here, dear students, due to the development of net internet facility in India, specifically government of India is also focusing on digital India and population, lot of there is a huge population, nearly 130 crore population. So these are all opportunities for you to start or to become an entrepreneur. So majorly you can focus on entrepreneurship skills and you can start like medical stores or e-pharmacies or internet pharmacies or likewise opportunities you can find out. So, shortly, government also drafting a plan for online pharmacy. So, how the licensing procedure should be, and so they are preparing a drafting plan. So here, lot of opportunities are there to start and to become entrepreneurs to the pharmacy practice. So you can take data like patient data like diabetes and what are the different diseases. So where you are going to start.
Excuse me, sir.
So in
Sorry to
that
interrupt
area,
you. Excuse me, sir. Sorry to interrupt you. Your volume is fluctuating, sir. Your volume is fluctuating. Sometimes it is hearing us like low and sometimes high. Can you please try without earphones once, sir? Because the participants are unable to hear sometimes. Or put your mic near to your mouth. The voice is fluctuating, sir. That's what. Sir has left, I think.
We are waiting for our speaker to join. He is having some network issues. Kindly bear with us and stay connected. He will be joining soon. Till our next speaker joins, I would request Paresh sir to kindly have a word with our audience, our viewers, and let them know what are the benefits of joining PharmaNest, and if he has some message to our viewers.
Yeah. So good evening to all who are joining with us, because we have technical some difficulties with our speaker Venkteshwarulu, and Pharmanest event we are organizing. So you must all be aware about this Operant Pharmacy Federation. So Operant Pharmacy Federation, our aim is that to help students and researchers to learn the latest development in the pharmaceutical domain. At the same time, our aim is also that not only you learn about the latest development, but you can also develop your skills. With these in mind, we are organizing many events throughout the year, and throughout the year we are also receiving very good responses from the students as well as from the researchers. And you can see that how many academic partners we have joined with us, that we have around 25 plus colleges and different universities joined as an academic partner. And these academic partners shows that their faculties are also taking interest, and they are also supporting to our event also. So for the students and researcher, it is more important to develop their skills and to know the latest trends whatever is going on in the pharmaceutical domain. With this aim, we are bringing Pharmanest 21 event. So under this Pharmanest 21 event, we are organizing many online competitions, as well as later on we will also plan to organize offline events. So online events, as it is mentioned in our website, you can visit to our website, that is opf.org.in/pharmanest. So if you go there, and you can see that we are going to organize many events like scientific presentations, like how to present a scientific paper, as well as how to present a poster, etc., and blog competitions. And we also have video competitions, and we are also having extempore competitions, where immediately you have to speak about the any one of the topic. Apart from this one, we are also bringing eminent speakers from the different field. From April 26, we have started webinars organization, and under these webinars we have invited many experts in the pharmaceutical domain as well as in the intellectual property right domains. So we are helping students, and we are assisting students to develop their skills. So today also we have with us Dr. Pillai, and Dr. Pillai also share his research and his topic about the machine learning, how it's important in the drug discovery, because machine learning and artificial intelligence is also helping for the drug discovery. Earlier also we have heard about this topic from the different speakers, and we also heard about the biosensors yesterday. So I encourage all the students and all the researchers for go ahead and do the registration for this pharmanest event and receive the latest development from our event, and you will receive, whatever the webinars we are organizing and whatever the different competitions we are organizing, and other news also you can receive it. So we have with us, today, Dr. Girinath Pillai. Dr. Pillai is with us.
Yes, sir. Sir is there. Maybe sir is on mute, I think. Sir.
Okay, so no problem. So our goal is that to upskill the students as well as the researcher. With these in mind, we are bringing webinars, and you can see that we are also trying to keep, means we are not taking any fees very high. We are just taking some token money, that to make sure that whoever is registered, they are seriously take part for the events. That is our main motto for this one. So with this
Yes.
mind, I would like to ask, the moderator of today, Srivalika and Sweta, to conclude the session. And if any message is there, you can please go ahead.
Thank you, sir. Thank you so much for an insight regarding Pharmanes 21, so that participants can get clear understanding regarding the conference. So we are still waiting our next speaker to join.
Um, Hello,
Participants.
Because
everyone.
we can organize later on, as we have technical difficulties. So
Yes, yes, sir. Yes, we can organize. We can put sir session later, sir. We can arrange later. And participants, you can post your queries in the chat box. I think there are no queries, sir. Okay, meanwhile, we'll have a glance at the feedback link. We'll share the feedback link now so that the participants can have a check into it. Yes, feedback link is shared on the screen. Participants can access the feedback link from the screen. Yes. So participants still having any queries regarding the Pharmanest, you can write us to pharmanest@opf.org.in regarding any event. You can write us. So this is the session for today. We can schedule our speakers, CDS CO, Mr. Venkteshwarulu sir's session on some other day. So we are going to conclude the session. Yes.
so have a
Okay,
wonderful
I have
evening
an
to
important
all the
announcement.
participants. Yes, yes,
Yes,
Sweta,
ma'am.
please.
Am I audible? Yes, sir. I
Yes.
have an important announcement regarding the dates of competition. The new date for competition will start from 21st of May 2021. So I request all the participants to get ready for the competitions, and all the best to everyone who has participated. And if anybody is still interested and not participated, the registration links are still open. You can just visit the Permanence website and do register with us to win over 10.51 lakh prize, 51 rupees prizes, and also an opportunity for international exchange program by Open Pharmacy Federation. And I also want to thank all the viewers to bear with us and stay tuned. It was an interactive session, and the questions were interacting. Hope we cleared all the questions, and hope you liked the sessions. Thank you, everyone.
Thank
Thank
you.
you,
Thank you.
everyone.